Hi, I'm Colin Tang. I'm a rising senior at the University of Delaware, and I spent my summer with the Hoadley lab researching coral restoration. Corals, as you may know, are the lifeblood of tropical reefs. With the help of their photosynthetic algae, they provide food, shelter, and many other ecosystem services to the reefs they inhabit. Unfortunately, corals are seriously under threat with the rapid acceleration of climate change in previous years. When corals are exposed to highly stressful environments, they expel their algae and become bleached, losing much of their functionality. The coral I focused on this summer is Parietes cylindrica, an Indo-Pacific coral that is a focus for coral restoration works. Not only does it grow fast, it also is relatively thermally tolerant, making it a useful species for understanding restoration. Cylindrica comes in two color morphologies, or color morphs, yellow or brown. My goals this summer were to quantify and see if they had two different thermal tolerances, and also to find out why they were two different colors. You can't really study a Pacific coral in Mobile, though, so I traveled to Fiji, as you do. In Fiji, we collected samples from unique coral colonies across various sites along the coral coast. Those samples were further processed into fragments that were about this size, and we had a total of 3 of them. 1 for phenotypic analysis using our custom-built fluorometer, and 2 to be ran in the sea bass, pictured here. The rest of the coral was clipped and stored in DNA Shield for downstream genotyping. The sea bass is a tool that we use to artificially expose corals to stressful simulations. There's one run with 2 treatments, one that was heated to a stressful temperature of 36 degrees Celsius, and one that was controlled and kept at an ambient temperature of 28 degrees Celsius. After the runs were completed, we took endpoint measurements of reflectance and FEFM. Back in the lab, we converted reflectance to absorbance, which I used to create these absorbance profiles of the 2 color morphs. On your left, you have the yellow morph. On the right, you have the brown morph. And for both graphs, the blue represents the control treatment, the red represents the heated treatment, and the dotted line denotes our area of interest, which is the chlorophyll A peak absorbance, the main photosynthetic pigment found in the algae. Absorbance is one of the metrics that we use to represent photosynthetic ability. If we imagine the coral as a factory, absorbance tells us how many workers there are in that factory. To zoom in on that chlorophyll A absorbance peak, I made these bar graphs. And as you can see, between the 2 color morphs, again, yellow on the left, brown on the right, there's not much difference between the 2. The controls are very similar, and the heated treatments are both very similar. Another metric we use to estimate photosynthetic ability is FEFM, which tells us photosynthetic efficiency. Back to the factory, this tells us how strong each of the workers are. And much like absorbance, there's not much difference between the 2 color morphs. You can see the 2 treatments there. My final graph is this phenotypic dendrogram, which tells us how similar the algae inhabiting each coral is, with, again, brown being represented by brown and yellow being represented by yellow. This is based on our phenotyping data, and it clusters the algae based on how functionally similar they are. If the 2 color morphs, if their algae were different, you'd likely see a large brown cluster and a large yellow cluster. However, as you can see, it's pretty well intermixed between brown and yellow, showing that they are functionally the same. And so what does that mean for my questions? To answer the first question, do they have different thermal tolerances? The answer is no, not really, which is shown to us by these endpoint measurements. And to answer the second question, why are they different colors? We still don't actually know. But our best guess is that it's something to do with the host, based on how similar the algae is. Our final question then is, what's next? For Parietes cylindrica specifically, there's still more metrics to be run. We can compare their GFP profiles or do a DNA analysis. For coral restoration overall, Parietes cylindrica is just one of very many species. There are still more species to analyze, color morphs to analyze, and much more to discover in general. Thank you.